Hello, welcome to CET Help 25. I'm Sri Lakshmi Nair, and today I'll be explaining the topic of post-fertilization events. The post-fertilization events are occurring after the double fertilization, which was explained in one of the videos. So, what mainly happens here is the development of endosperm and the development of an embryo. So what is an endosperm? Endosperm, like mentioned before, is the primary endosperm cell, which was formed after the fusion of the polar nuclei with one of the male gametes of the pollen grain. So the primary endosperm cell, which is triploid cell, that is the nucleus contains three ends, three haploids, fusion of three haploid nucleus. So the primary endosperm cell, it divides mitotically. So the division of cells occurs in a mitosis, and it forms the primary endosperm tissue. So that is what happens here. There is the formation of primary endosperm tissue by the division of primary endosperm cells in a mitotical manner. This is clear, I hope. And this actually occurs before the development of the embryo. So there is a little development of endosperm before the embryo or the zygote starts developing. And this is very important because the endosperm actually is the vegetative or the nutrition providing cell. So once the embryo, that is the reproductive part of the flower, the zygote, it starts developing, it requires nourishment. It requires some food reserves which is provided by the endosperm. So thus the development of the endosperm before embryo development is very necessary, or it is very vital. Now we will explain the embryo development. So we had learned the formation of a zygote, that is when a male gamete, it goes and fuses with the egg cell of the megaspore or the ovary. So the zygote then it develops into the embryo. The zygote, it also undergoes mitotical divisions to develop into the embryo, and it develops at the micropylar end of the ovule. The micropylar end is the opening of the ovule, and the zygote development usually takes place in that area. So, in general, there are two types of embryos. That is, one is a monocotyledon embryo, next is a dicotyledon embryo. So, monocotyledon embryo, it consists of only one cotyledon. Common dicotyledon embryos are generally very common embryos, and they provide a better nutrition to the development of the seed or the maturation of the seed. But in the initial stages of embryo development, both the stages are very similar in both monocotyledon and dicotyledon embryo development. The zygote, this is the zygote. It undergoes mitotical division. Then it undergoes a structure like this, which is called a globular embryo. So it forms a it undergoes mitotical division to form a cluster of cells like this, which is a globular structure. Further, these cells undergo mitosis, and this heart-shaped embryo, it comes under the dicotyledonous embryo. It forms into two cotyledons, and this is the initial stage of the development. This further undergoes mitotical divisions to form the mature embryo, and this is an example of a dicotyledonous embryo. So that is the topic of post-fertilization events. I hope you understood, and I would like to conclude this topic. If you like the video and if you understood the topic, please click the like button and add your comments below, and also share it with your friends. Thank you for watching.